Hi students, welcome back to my channel. Once again, I am Ma'am Dea, and today I will be teaching you the central dogma of molecular biology. Last time, I discussed the difference between DNA and RNA. So kung hindi mo pa napapanood ng video na yon, pwede mong balikan ang video gamit ang link na makikita mo sa upper right part ng iyong screen. So ngayon makikita natin sa inyong screen na ang central dogma of molecular biology ay may tatlong steps. Meron tayong tinatawag na DNA replication, and then we have transcription, and finally we have translation. Later on, I will give you some examples where you can practice para mas maintindihan ninyo ang pagkakaiba ng bawat step. What is the central dogma of molecular biology? The central dogma describes the flow of genetic information in cells from DNA to messenger RNA and to protein. This diagram shows the summary of all these three processes. Bakit protein synthesis? What is protein synthesis in the first place? Protein synthesis is the process of making proteins. Bakit natin kailangan ng proteins? Because proteins are the executor of cell life activities. We are made of cells, so for us to be able to function properly, our cells must be able to produce proteins. Remember, proteins are made up of amino acids. Amino acids are the building blocks of proteins, so without these amino acids, there will be no proteins. In your screen, makikita din natin na before amino acids become proteins, they become peptides. So peptides are short amino acid chains. Mamaya makikita natin kung paano na-form ang peptide at ang protein. On your screen, you can see the summary of the differences between RNA and DNA. Again, this was already discussed in the previous video lesson. So, RNA stands for ribonucleic acid, while DNA stands for deoxyribonucleic acid. Both types of nucleic acids are made up of what we call nucleotides, which consist of a phosphate group, a sugar, and a nitrogenous base. Basically, RNA and DNA have 4 different types of bases. Nagkakaiba lang sila sa uracil at saka sa thymine. Tandaan natin na itong 4 na nitrogenous bases na ito ay meron silang kapartner. Hindi pwedeng mapalitan ang kanilang kapartner. Adenine pairs with thymine, while cytosine pairs with guanine. In the case of RNA, adenine would pair with uracil kasi sabi nga natin kanina, wala ng thymine sa RNA. In the central dogma of molecular biology, it is also important for us to identify the different types of RNA. We have 3 types. We have the messenger RNA or mRNA, we have the ribosomal RNA or the rRNA, and we have the transfer RNA or the tRNA. Now, what is the difference among their functions? The messenger RNA or the mRNA carries genetic codes from DNA or nucleus to ribosomes. If you remember in the previous video, we have said there that DNA cannot go out of the nucleus. It needs RNA for the message to be transferred from the nucleus outside the nucleus. So, kailangan niya rito si mRNA. Kaya siya tinawag na messenger kasi siya yung tagadala ng information. Ribosomal RNA consists of 80% of the total RNA in the body. They primarily stay in what we call the ribosome, and these ribosomes are found in the cell. Finally, transfer RNA or the tRNA, they transfer amino acids during protein synthesis. So mamaya malalaman natin kung ano ba yung amino acids na tinatransfer nila. punta na tayo sa DNA replication. So on your screen, you can see a summary of what happens in DNA replication. Now, what do we need to remember for DNA replication? In DNA replication, two identical DNA molecules are produced. Replication is similar to duplication. So, ibig sabihin, from one strand of DNA, it would become two strands, which are identical to one another. However, the process is semi-conservative. So, mamaya, i-explain ko sa inyo kung ano ibig sabihin ng semi-conservative. So, in this process, we have what we call key players. So, these key players are enzymes. When we say enzymes, these are molecules that speed up the rate of chemical reaction. So, you need these enzymes in order to do something to DNA. So, remember, in our discussion of the difference between DNA and RNA, the nitrogenous bases in DNA are glued together using what we call hydrogen bonds. And it is nearly impossible for hydrogen bonds to be cut or for the nitrogenous bases to be separated from one another without using enzymes. So, ano-ano ba itong mga key players na ito? So, itong pictures na ito ay kinuha ko sa module namin sa Grade 10 Science, Module 4 of Division of Caloocan. As you can see, there are 4 key players in DNA replication. We have helicase, we have primase, we have DNA polymerase, and we have ligase. So, para mas maintindihan natin kung ano yung nangyayari dito, I will show you a simulation of what happens. Again, for DNA replication, you start with a parent strand and you come up with 2 daughter strands which are identical to each other. Makikita natin that DNA splits into 2 separate strands. What causes the split? It is the enzyme helicase. The helicase is the unzipping enzyme. So, ang ginagawa ni helicase, parang ginugupit niya yung pagkakadugtong ng mga hydrogen bonds in between the nitrogenous bases which are adenine, thymine, guanine, and cytosine. And then, what happens? So, after that, we can see that the different nitrogenous bases attach to each strand. So, syempre, yung mga bases na yan, hindi niya yan mahahanap agad. Kailangan merong mag-start ng pagdidikit. Kailangan merong mag-signal kung saan magsisimula. And that enzyme is called the primase. That's why we called here primase as the initializer. It gives or serves the starting point for DNA replication. Makikita natin na kumakabit yung mga nitrogenous bases doon sa strands ng old DNA. So, makikita natin yung old DNA, itong nasa baba at saka itong nasa taas. So, yung nasa gitnang part, okay, so these are new strands. Ngayon, syempre, yung nagbi-build ng mga nitrogenous base dyan para maka-form ng bagong DNA strand ay yung tinatawag natin na DNA polymerase. That's why it is called the builder. And of course, kailangan mag-glue or madikit yung mga nitrogenous base na yan. And we call that gluer the enzyme ligase. So those are the functions of the 4 enzymes which we call the key players in DNA replication. So once the pairing is done, what happens is that the DNA coils back again, and afterwards there will be now 2 copies of the original DNA. here I have a short DNA strand which we will be using as an example for DNA replication. So again, DNA is double-stranded, so you can see a strand on the left and a strand on the right. I already placed here the different nitrogenous bases and placed the correct pair. So cytosine would pair with guanine, thymine would pair with adenine, and vice versa. What will happen during DNA replication? So during DNA replication, the DNA unzips. So what happens is that new nitrogenous bases would attach to the strands that unzipped. Okay, so here I use different colors for the old strand and the new strand. And then, of course, magkakaroon ng mga nitrogenous bases yan based on the pairings that we have said a while ago. Now, on your papers, can you try filling up these blank parts? What do you think would be the nitrogenous bases that will appear here? Okay, so let's start with this. That would be G because cytosine would pair with guanine. Okay, this one would be A because thymine would pair with adenine. Okay, and then this would be letter C because guanine would pair with cytosine. And we will do the same process all over again also for the other strand. So we have T, T, A, C, T, G. Now, after DNA replication, we have two daughter strands, this one and this one. And if we compare the base pairing with the original strand, we can see that they are just the same. We have CG, TA, GC, AT, AT, TA. The only difference is that one part of each daughter strands or daughter DNA would be coming from the old or the original strand, and another part would be a new strand. Again, this is what we meant by saying semi-conservative during the start of our discussion. Okay, so now let's go to RNA transcription. What happens during RNA transcription? During RNA transcription, a region of the DNA that was replicated a while ago will be converted to mRNA. Sabi nga natin, si DNA, hindi niya kayang lumabas ng nucleus. So, kailangan lumabas or mailabas yung message. Kailangan ma-code siya. So, yung mRNA, dun na siya papasok. Again, ano ibig sabihin ni mRNA? mRNA stands for messenger RNA. On your screen, I am now showing you the simulation of RNA transcription. So, una step dyan, mag-a-unzip si DNA. Let's now focus on just one DNA strand. Mag-a-unzip siya inside what we call the enzyme RNA polymerase. Sabi nga rito, the DNA unravels and then nucleotides of RNA join to one strand of DNA. So this process of joining still follows the base pairing that we have discussed in the previous videos. However, since this strand is RNA, you will expect that it would attach uracil to adenine instead of thymine. In short, dito, wala na dapat thymine sa lalabas na messenger RNA. So, once na nakopy na niya, nakuha na niya yung kailangan niya, lalabas na si messenger RNA sa nucleus. at pupunta na siya sa next step. So again, what is important to take note here that this time adenine should pair with uracil. Still, there would be thymine here that would pair with adenine, but once adenine is present in DNA, it would pair to uracil. So for us to have a concrete example, I will be showing you the PowerPoint. Now, this is the original DNA that we have used a while ago as an example for DNA replication. Okay. Again, for RNA transcription, what will happen is that the strands would unzip again, and then mRNA will copy the coding strand. So, let's say the right side is the strand that is to be copied. You just simply have to identify what base complements the bases already found on the right. So for G, the complement is C. Okay, for A, for adenine, the complement would be U. Okay, for C, we have G, T, we have A, T, we have A, and A, we have U. So this is what I have been stressing a while ago, that this strand is already mRNA. The green part is from DNA. So since the yellow part or the orange part is already mRNA, dapat walang letter T dyan. So, adenine would pair with uracil. But still, if you have thymine here on the DNA side, it would pair with adenine. What will happen now to mRNA? mRNA will leave the nucleus and it will go somewhere. Okay, any questions? After the process of RNA transcription, DNA zips back together. Didikit ulit sila. Finally, let's go to the third process, which is translation. What happens during translation? Remember from the previous process, mRNA copied the information from DNA and it went out of the nucleus. It will now go to the cytoplasm. So what happens during translation is that information in the nucleotide base sequence of mRNA is used to dictate the amino acid sequence of a protein. Again, the goal of the central dogma is protein synthesis, and for our body to create proteins, it should start with the building blocks called the amino acids. O si mRNA, pupunta siya kay ribosome, doon na siya i-meet ni tRNA para magkaroon ng amino acid sequence. So let's try to show what happens during this process. So on your screen, you can see again the simulation. So the one you can see on the lower part of the screen is the messenger RNA. It goes to the ribosome and you can see here na merong mga dumidikit-dikit sa kanya. Okay? And these are your tRNA. These tRNA have a specific amino acid attached to them which corresponds to the mRNA or the messenger RNA. Mamaya i-discuss ko kung ano yung tawag sa mga yan. We can see here that each tRNA would have the amino acids, and once na matapos niya tong buong strand na yan, nagdudugtong yung mga amino acids to become the peptide chain, and eventually, if you have a lot of that, you will be having the protein. So let's watch the whole process. So makikita natin na dumadami na at humahaba itong mga amino acids which will become peptides and eventually the protein. Until matapos po si mRNA, ayan, magkakaroon na tayo ng buong protein. Now, if you will notice, the mRNA is being coded in groups of 3. Okay, may tawag dyan sa mga groups of 3 na yan. Going back to our PowerPoint, here I am using the same mRNA strand that went out of the nucleus in our example in the previous slides. So, mRNA is divided into groups of 3 which we call codons. This mRNA strand will be translated by what we call the transfer RNA. I used color red to designate the tRNA strand. So, kung sa mRNA meron tayong tinatawag na groups of 3 na codon, sa tRNA, ang tawag natin dyan ay anticodon. Alright? The same set of rules would be followed for the base pairing. However, take note that mRNA and tRNA are both types of RNA. So, there should be no letter T or thymine this time. That means that C would pair with G, okay, and U would pair with A. Can you finish the remaining letters? Okay, so G would pair with C, A would pair with U, A would pair with U, and U would pair with A. So, tapos na po ba tayo? Hindi pa. Because these sets of nitrogenous bases must be translated into a specific amino acid. We need the amino acid to be able to come up or produce a protein. Ngayon, paano natin malalaman yung amino acid? We have what we call a codon table or a codon chart. You will be using the codons found in the mRNA to find the name of the amino acid. Here we have around 20 amino acids and you will just be reading this side first and then the second letter you will be looking at the top part and then for the last letter you will be looking at the right side. For example, in the mRNA you can find the codon AAU. So, how do we read that? First letter is A. Okay, here. Ibig sabihin, mag-focus lang tayo sa part na to. Then, second letter is A. So, tayo pupunta. A. So, ito. Dito lang tayo pupunta. And then, you find U. Okay, this would be AAU. AAU corresponds to the amino acid asparagine. How about GCC? Can you try finding the amino acid corresponding to GCC? If your answer is alanine, that's correct because we have G, C, and C. This is GCC, alanine. How about CUU? If you answered leucine, that is also correct. So, C-U-U. Leucine. So, now that you have been able to understand how to read a codon table or a codon chart, we can now use it for our example. Again, remember that we have to read the codon, not the anticodon. So, even though you are able to identify these anticodons, these are not the things that we will be looking for in a codon table. We have to focus on the codons found in the mRNA. So first, let's find CUG. We have C, U, and G. CUG corresponds to leucine. Okay. How about the next one? Can you find it? So find the amino acid that corresponds to the codon AAU. If your answer is asparagine, that is correct. So why is it asparagine? You have AAU. Asparagine. So in our example that we have been using since DNA replication, we only used a short strand. Kasi for the sake of just showing you an example. But then in reality, this would be a long strand. So you will be having a lot of amino acids because you need these building blocks in order for you to come up or to produce proteins. And the process continues. If you will notice, we have AUG, which is methionine, the start codon, and we have three types of stop codons. We have UAA, UAG, and UGA. Anong relevance ng mga to? Usually, the start of translation is AUG. It signals the start. That's why it's called start codon. So usually, it starts with AUG. And then, until such time that the tRNA reads a stop codon, which would be UAA, UAG, UGA, dun lang siya titigil. So, once na ma-read niya yung stop codon na yan, mag-i-start siya ulit ng process. Okay, so, I hope I was able to give you a simple discussion of the central dogma. Apologies if I frequently used Filipino in my discussion because I'm trying to discuss the concepts in the simplest way possible. If you have questions, don't hesitate to post them on the comment section below. Now, if you are new in my channel, please don't forget to like and subscribe, and you can share this to your friends. So that's all for today. See you in my next video lesson. Bye!